you very much, Deborah. Well, I'm delighted to be here. I could never have imagined I would end up becoming a professor of anything, actually. When I started my journey in education, I had no idea what I wanted to do when I was a teenager. My dad was he left home as a teenager to join the RAF, and then spent my whole childhood moving from school to school, house to house. So in fact, when I was 14, I went to 4 secondary schools. So 1 in Germany, 1 in Kent, and 2 in the Midlands. So my education was quite disrupted, and it was very fortunate that I found maths and physics quite straightforward, and I think that rescued me really, because I wasn't expecting to do A-levels. Hadn't really come from a background of a family that had an education, and I was fascinated by wildlife, and I wrote to the World Wildlife Fund asking if I could become a warden on a safari park in Africa, and they politely wrote back and said, sorry, no, we only employ locals. So I then got really into martial arts. In fact, I used to train about 6 nights a week, 3 hours a night, and when I was 16, 17, I had the opportunity to become a martial arts instructor. But I carried on with my A-levels, and my friend, who I trained with, he did go on to be a martial arts instructor, and then went on to become a personal trainer for Hollywood stars like Angelina Jolie and Tom Cruise. But I think I made the right choice in the end. I'll be coming back to Angelina Jolie later, actually. So I was at Orpington College as a 16, 17-year-old, and in the library, and I opened this book on microbiology, because I didn't really know what microbiology was, and saw these bacterial cells with little circles of DNA called plasmid DNA. And I was fascinated by this, so I looked at guides to university and found courses and applied, and to my surprise, got offered interviews and went to Bristol for an interview and really liked it. So I decided to go to Bristol University. Now, I didn't think I'd last very long because I'd never met anyone from university before. I thought they were a kind of different species. And in fact, a lot of the people at Bristol, if those of you have been to Bristol, there's a lot of posh people, posh students from public school. And in fact, my first girlfriend there wasn't impressed by my vocabulary. And so for my birthday, she bought me Roget's Thesaurus and Colin's English Dictionary. And then we split up. And I couldn't work out why I was always stopped at customs. I can see why I was always stopped at customs when I was a student, a young adult. And the first 2 years were a bit dull because it was all theory, really. And I had an opportunity to get a 3-month temporary work permit to go to America. And it was just a stunning experience, and I can't believe I managed to survive it because I ended up hitchhiking around Nevada and California, sleeping under the stars in Death Valley, sleeping in car parks in Los Angeles, sleeping on the beach at Big Sur. And I worked at a casino in South Lake Tahoe and got evacuated one morning. Someone had wheeled in a 1,000-pound bomb disguised as a mainframe computer and asked for $3 million, otherwise he'll blow it up. And apparently the FBI turned up at the wrong place to deliver the money, and it went off. So that was a close shave. So I came back to England, just made it back, and kissed the tarmac, and entered my final year. It might be difficult to see, but I really had an epiphany on the 3rd of November 1980. I dug out this color Polaroid picture where I started my laboratory research project, actually doing things with my hands, and it was a real epiphany. And I found this old photo, which is actually the medical school at Bristol, and there's a ray of light coming down onto the lab where I actually took that photo. And I just realized that, you could actually do something that you really enjoy and get paid for it by doing research. And it was a real epiphany for me. So I decided from then on, I was going to do research. I had no plans to do research or to do a PhD. I didn't know what I wanted to do, but it all fell into place then. And this plasmid was actually the very first resistance plasmid isolated from a Burns patient in Birmingham, which conferred resistance to all the commonly used antibiotics. So I decided that I wanted to work in something to do with antibiotics. And I graduated in my first suit, which I picked up the day before my graduation, with my mum and dad, and got cleaned up, and went off to the John Innes Centre to study streptomyces. Now, streptomycetes were a very odd bacteria, but the amazing thing about streptomyces is they produce almost all of the clinically used antibiotics and many other bioactive molecules like anti-cancer drugs. And I went for my interview with Sir David Hopwood, who was the founder in the 1950s. Nothing was really known about bacterial genetics. He decided he wanted to work with this bacterium, and he chose this one, Streptomyces silicolor, which is the color of the skies, because he liked the color. He was very fortunate. It turns out that blue pigment is a really important antibiotic, and he got lots of Nature and Science papers out of studying that, the genetics of that. But another reason I fell in love with it was the smell. Streptomyces produce geosmin, and I've actually brought some Petri dishes with Streptomyces silicolor on it, which you can all have a sniff during drinks. It's a wonderful smell. It's actually responsible for giving earth the smell of earth. It's a fragrant, earthy smell. And the human nose is very sensitive to it. And it's just when I woke up the following morning in Bristol, I opened my plastic bag and I got hit by this waft of Streptomyces, and I thought, right, I'll do my PhD on this. So I got cleaned up, looked more respectable, still got stopped at every customs. And when I went to Ireland as a young adult, they actually took me into an interview room. I said, why do I always get stopped at customs? He said, I'm afraid you've got terrorist eyes. It was nothing to do with the hair and the beard. It was the eyes. But apparently they've kind of drooped. I don't get stopped anymore. So Streptomyces, this is the edge of a colony of Streptomyces. It's nothing like a normal bacterium, which is like a shiny single-celled colony, a single-celled organism which produces a shiny colony. It almost looks like a fungus, but it's a bacterium. And I'm only going to give a brief tour through some of this, but antibiotics, almost everybody knows about antimicrobial resistance and the global threat. This is a poster produced by the Cross-Council Initiative. AMR is one of the world priorities, resistance to antibiotics. And this is a kind of timeline. And antibiotics really were discovered and applied during the Second World War. They saved loads of servicemen. And then that was really the golden era of antibiotics. But as antibiotics started being developed, within a year, like methicillin, a derivative of a penicillin, within a year, methicillin-resistant staphylococcus were coming out, these so-called superbugs, within a year of its use. And it took about 30 years to develop a completely novel antibiotic called daptomycin, which was in 1987. It's taken another, 30 years to actually develop, to identify another completely novel one, which is very new. It's called Teixobactin. Don't know whether you can see it, this small text. But daptomycin is a class of antibiotic that's used. It's last line of defense antibiotic. It's given intravenously if a patient is resistant to vancomycin, if the bacterial infection is resistant to vancomycin. Here are some examples of some products from streptomyces and their close relatives. They produce, if you have a good enough screen, you can find something that will have whatever biological activity you want. I mean, there are immeasurable numbers of species of streptomyces. Each one of them produces about up to 50 different natural products unique to that species. It's a very rich source of natural products, but including anti-cancer drugs, clavulanic acid, which is a major product of GSK, UK company, GlaxoSmithKline, is a streptomycete product, and daptomycin, which is, I mentioned before, and it's fortuitously very similar to an antibiotic that I worked on in the model organism, streptomyces. And this natural product was made freely available to the developing world to treat river blindness. It was discovered by a Japanese scientist who was awarded the Nobel Prize for ivermectin. So if you're going to use genetics, I was interested in molecular genetics. If you're going to use molecular genetics to exploit streptomyces to make new antibiotics, you have to understand what genes look like. And nothing was known about genes in streptomyces. So we had to go back to develop methods to look at what the gene structure was like, whether they were, how they were transcribed, how genes were controlled. So I need to introduce some basic molecular biology, really to provide a context for the rest of my talk. So pretty much all organisms, except for a few viruses like influenza virus, which have RNA genomes, all other life has DNA as the heritable material, and it's invariably a double helical structure. And remarkably, it's built only of four building blocks, A, C, G, and T. And the A always base pairs with T, and the C always base pairs with G. It produces this beautiful right-handed double helix. That means if you go like that with your right hand, if you want to draw one, you draw it that way with your right hand. And there are 10 base pairs for every complete turn of that double helix. And this is looking down on the DNA molecule. It's really beautifully packed in. And the DNA is an incredibly stable structure. And it's these bases in the center of this helix which actually confer the properties on that DNA. And so just a bit of introduction to gene expression, because I'm going to be talking about transcription and translation later in the human studies as well. Genes are actually bits of DNA which have very defined starts and stops on the double helical DNA. But one of those strands is actually transcribed into another nucleic acid called messenger RNA, and that means it's become active. So the DNA itself is static, but genomes are dynamic, and that's because you have to switch genes on and off. And that's what I'm going to be talking about mostly today, is the dynamic nature of the genome. And once that RNA is made, that intermediate is then translated to make the proteins. So most genes code for proteins, and they're translated by ribosomes, which load onto this RNA, traverse the RNA, decoding the RNA in a triplet code, which I'll come on to shortly, to produce proteins, which are the final products of the gene. Now, the only reason I'm putting up this slide is just to emphasize that every cell of an organism contains the same genome, and that's in our cells, it's contained within a nucleus. And so every cell has the identical genome, which is very highly packaged inside the cell. So if you could open out the DNA, it would actually extend for about 1 meter. The amount of DNA, chromosomal DNA, just in one human cell would stretch out to about 1 meter. So just to keep on the introduction to genomics, I thought I'd put the human genome in here as well. So our genome is made up of 46 chromosomes. So we get 23 of those from our mother and 23 from our father, from the egg and the sperm, respectively. And you can see there are 2 of everything. So chromosome 1 is the biggest one. Chromosome 22 is the smallest one. All of us have these. Females typically have 2 X chromosomes, and males have 1 X chromosome and this pathetic little Y chromosome here. And essentially, the reason because we get a chromosome from each parent, it means we have 2 copies of every gene in the genome. And it's a lot of DNA packed in. It's actually about 6 billion base pairs, 3 billion from the mother, 3 billion from the father. And a genome, our genomes have tens of thousands of genes. And as I mentioned, they're dynamic. They're not active all the time. They're only brought in to function when they're needed. So my PhD was concerned with establishing methods for studying gene regulation with Sir David and with Keith Chater, who was my principal supervisor, but I worked in David's lab. And so this was, because until we did this study, we didn't know what genes looked like. So we defined what genes looked like. And I finished my PhD, that's me circled there, and I was fortunate enough, David let me stay on for a year as an assistant, which allowed me to write up my PhD and actually write my first grant application. And also to be an instructor on a European Molecular Biology Organization course on streptomyces in 1985. Because for personal reasons, I wanted to go back to Bristol. And I actually I went back to Bristol and managed to persuade an academic in the biochemistry department to provide a lab if I could write a grant. And he was very generous and altruistic. He said, well, if you write the grant, I'll put my name on it and you can come and work. And his name is Nigel Brown, who's a wonderful scientist who's had many roles, Director of Science and Technology at BBSRC, the Head of Science at Edinburgh, and President of Microbiology Society and many other roles. He wanted to come today, but he's actually at the Royal Society of Biology dinner because they're handing over presidents tonight, and I didn't realize it clashed with that. And he was actually a co-author on the very first paper with Frederick Sanger on the first genome to be sequenced, which is a bacterial virus, FIRX174 genome. So I continued these studies in biochemistry in Bristol to define genes, and that really set the scene. So this is a kind of example of the kind of techniques we would use. The only important thing is each one of these fragments here differs by a single base pair. And this is measuring transcription, so the stronger signal means there's more RNA for that gene, and these are two different endpoints of RNA. So the stage was set then to move on. I was very fortunate to get a position straight out of my postdoc as a lecturer at UMIST in Manchester, which doesn't exist anymore. It was actually, I think it was ranked in that our research assessment exercise. It was ranked 5th or 6th in the country as research institutions, and it was ranked very highly in biological sciences, and with a fairly small department of 21. And in 2003, we merged. Well, I left a couple of months before it merged with the University of Manchester, and so it was just it was a really exciting, vibrant place to do science, and I started a collaboration with David Hopwood and others to actually identify the genes for this really powerful antibiotic, which is related to daptomycin, which kills superbugs. And we managed to clone that, all of the genes for that. And Jason Micklefield, a chemist from across the road, came knocking on my door. I've just seen this paper on CDA. Why don't we work together? And that was my first interdisciplinary experience of getting together with a chemist, because my chemistry was awful. Because my education was so disrupted, I couldn't even do chemistry during my schooling. I did maths and physics, statistics and biology instead. So we got together. My group in Manchester, we characterized all of the genes. And the great thing about antibiotics, it doesn't matter what, there's 39 arrows there. Those 39 arrows make the antibiotic. And the amazing thing about nature in streptomyces is everything you need to make an antibiotic is clustered physically in one space. So we can actually go in and take it out and move it into different backgrounds and manipulate it. And so we dissected the function of every one of those genes. And this big yellow arrows, this is an enormous monster of an enzyme. It actually contains 13,550 amino acids. is 1.5 million Dalton enzyme. It's enormous. And it's a production line. That yellow structure produces that giant enzyme. It has more than 30 individual active sites on it, and it takes one molecule and then it joins it to the next one, to the next one, to the next one. So it literally is just like one of these robotic production lines for making cars. It loads up one end, it comes out complete at the other end. They're remarkable. And that's fantastic for genetic engineering, because we can just go in and change whatever we want to change the structure of the antibiotic. And we managed to publish a major paper where we actually we defined the functions of all of those genes, and we made the front cover of this interdisciplinary journal in 2002. And just as an aside, just for academics here and the research excellence framework, just an example of how impact factor shouldn't always be used. So I submitted a paper. It's got a horrible title, a really complicated title, but how we could get DNA from some bacteria into streptomyces. And it got adopted worldwide in academia and industry, and it's my most highly cited paper as senior author, and 424 citations, and it's the lowest impact factor journal I've ever published in. Which is, so I agree with people when they say don't look at impact factors. So I'm just going to jump, this is fast forwarding to present day, simply so that I can just keep the genomics technology all in one place and keep streptomyces in one place. And I talked about transcription, but what about translation? When we're studying gene activity, we look at the RNA, and if there's a lot of RNA, we say there's a lot of gene activity. But there's another stage where you actually load ribosomes and make the protein. And there's a new technology has been developed called ribosome profiling, where we smash the cells open, we add an antibiotic, it's a streptomyces antibiotic, chloramphenicol, stops the ribosomes in their tracks. We smash the cells open, we treat them with nucleases, then we purify these little ribosomes which are trapped on the RNA. We then remove the ribosomes, and we're left with these tiny little fragments that got protected by the ribosome attached to it. And if we can then actually identify every single one of those and map them onto the genome, so we know exactly what bits of every RNA are actually being translated at the time that we smash the cells open. And that's given us a completely different picture of gene regulation, and that applies to bacteria and right through to humans. This technique is the latest genomic technique. And we got together with Byung-Kwan Cho and other people in Korea to learn this technique. So I went with Giselda Bucco, who's here, over to Korea and probably knocked 10 years off our life, working like 18 hours a day in their labs for a couple of weeks, and basically learnt this technique and managed to work out. From this study, we could show that translation played a key role in switching on antibiotics. So it's opened up a whole new way of manipulating pathways to discover antibiotics, enhance antibiotics. And on the basis of that international collaboration, we now have a collaboration with GlaxoSmithKline here in the UK, where we're applying this technology to their industrially important products. And just today, received the proofs this afternoon of a paper where we've applied. This is the first major paper with Brighton at the front and the front and the back of the paper on using this new technology. So now I'm going to jump from bacteria to humans. And the thing is, because I studied DNA and RNA, it doesn't matter what the organism is, because DNA is DNA and RNA is RNA. It's the same molecule, whether we take it from you or take it from a bacterium. It actually turns out it's a lot easier to work with human RNA than it is to work with bacterial RNA. So I got together with a team at Surrey. They have a really good clinical research centre, and they have a sleep research centre in there where they have 12 isolated bedrooms. So once someone goes in there, they don't know what time of day it is, they don't know anything. Everything's carefully controlled. And you can assess their sleep, wake, cognitive testing. You can take biopsies. And they have cannulas, and then there's a pump through the wall, and you can actually draw blood with automated machines to sample continuously. Well, not continuously, they bleed to death. But to sample like every 2 hours, every 4 hours, to analyze what's happening to gene activity. And this was a collaboration with the sleep researchers in Surrey. It was a very powerful, very strong sleep research team headed by Dirk-Jan Dijk and a number of others, including Malcolm von Schantz, who I continue to have a collaboration with on circadian disruption, where we're developing a network with a number of other people to apply genomics to circadian disruption and mental health. So this unit has 12 beds, so you could analyze 12 people. And this is the great thing about genomics. Because we know the genome, we have the whole human genome, we can manufacture DNA for every gene, and we can print it at very, very high resolution on a special surface. And so this what looks like a carpet here, represents every gene in the human genome. And then we can isolate that RNA, we can make it fluorescent, and then we can look at we can actually quantify the signal from every one of these spots, and it tells us how much that gene is active in that sample. And so you can consider genomes as like a huge cast in a dramatic play. Each cast member is a gene. They just, they come out when they're needed, do whatever they want to do. They go off, and then other cast members come on. So you've literally got the full cast there, and then you can see which ones are active in different acts. And so this was applied to sleep. And the only thing people are really happy to give you is blood. You wouldn't get far sampling other tissues on a regular basis. So we looked at blood, at gene expression in blood. And it turns out that to look at blood, what's going on in the blood actually reflects what's going on in other organs in the system. The blood pumps through all your organ systems. There's a lot of signaling. And actually, it turns out what we were finding in human studies, in the human blood, actually fitted what was being found in animal studies elsewhere, where they looked at gene expression in the brain and in the heart, in the liver. And we managed to get two major studies published in 2013 and 2014, which are, the citations reveal the impact of those papers, where we've actually been able to look at what happens to gene activity in the blood when you disrupt sleep. So insufficient sleep, people are running, I'm going to just go through this quite quickly, but essentially they come in and they just settle into their little isolated room, and then they're allowed to sleep for, in this case, they're allowed to sleep for up to 10 hours, and this goes on for about a week, and then they're kept awake. And then it's a double crossover, so they'll come back, and then they'll, so this one, then they'll be restricted, and they can only sleep, they're woken up after 6 hours. So they're allowed to sleep up to 10 hours, or they're woken up after 6. And to cut a long story short, lots of things happen when you do that, and there's lots of genes are activated, and they're dealing with stress, repair of tissues and everything, and lots of other key genes are downregulated following insufficient sleep. So, and that's providing a lot of insight into future studies of which pathways are affected by sleep deprivation and sleep restriction. And I'm convinced that all of this is going on in my blood transcriptome. It's called the transcriptome, the description of volume, because I haven't had more than 6 hours sleep for at least 2 weeks. And the other one was looking at missed time sleep, which is the jet-lagged shift work protocol, where when you go to sleep, well, when it's time to go to bed, melatonin starts to be produced by your brain. It signals it's time for bed, and you go to sleep. Here, the volunteers are actually shifted by 4 hours every day until after 3 days, they've been completely phase shifted by 12 hours. And if you monitor, this is all continuous monitoring here of blood over those little triangles of the monitoring. And the bottom line is that a lot of genes follow a circadian profile, a 24-hour cycle. Most of those collapse if you shift your phase. So here, each one of these rows is a gene. This is a 24-hour clock. So when you're asleep, these blue genes are down. When you're awake, these red genes are up. And these genes follow the opposite profile. And you can see after shifting 12 hours, a lot of that just collapses. And that explains why it's so unhealthy to basically shift your clock, because it messes up the natural homeostatic control mechanisms in your body. So this moves on to another interdisciplinary collaboration. I've just noticed that Sue Lanham-New is here. I'm delighted to see that Sue's arrived. And and this is on vitamin D. So Sue Lanham-New at the University of Surrey has studied vitamin D for many years, has a very high profile in this area. In fact, they were awarded the Queen's Anniversary Prize, I think, last year for their research and teaching in this area. And so I don't know how it happened. Why don't we combine and do some genomics of your vitamin D study? So vitamin D is made by your skin in the sun. But that's okay if you're at the equator. But if you're in Britain, or if you're further away from the equator, your skin doesn't really make enough vitamin D. It's the UV light which causes vitamin D to be manufactured in the skin. And if you put sunblock on or sun cream, that prevents it. And there are a lot of people for example, the Inuits, people away from the equator, who are not going to get enough vitamin D. And of course, people who wear burqas are covered up, they're not going to generate vitamin D. And people tend to think you get enough from the diet, but as you'll see, that's not actually the case. And so this study that was put together with Sue and with the food industry, as BBSRC Food Industry Joint Funded Program, to look at the relative efficiency of two vitamins. So you probably don't realize there are two types. Vitamin D3 is the real thing that our skin makes. Vitamin D2 is derived by shining UV light on mushrooms. And it was thought that they were equivalent. And it's been used a lot in supplements. So Marks and Spencer's brought out vitamin D enriched bread. That's vitamin D2 enriched bread, not vitamin D3. And so we get our vitamin D3 from the sun, but also from things like animal products like eggs and fatty fish, cod liver oil, milk. But it's important to bear in mind the type of vitamin D. So in this study, Sue put together this big trial. After doing a meta-study, which is very highly cited, this trial was funded by the BBSRC and the food industry to compare D2 and D3 at raising the serum level of a derivative of vitamin D, which is how your vitamin D status is measured if you have a blood test at the GP. And this study showed that vitamin D3 was significantly better at raising the level of your vitamin D status in your blood. While this work was being done and hundreds of subjects were being recruited, blood was taken from all of them and archived in the -80, which I'll come on to in a second. So this is actually the data. This was a fully randomized placebo-controlled trial. Those given D3, the serum, the vitamin D goes up. There's about 30 nanomoles per liter. It's in a later slide, is actually what's considered to be the limit. You're at risk of being deficient, vitamin D deficient, if you're below 30 nanomoles. You can see that those given no vitamin D dropped even further. This is in the winter time, so these experiments were done between October and March, where there's not enough sunlight to generate vitamin D from the skin. Vitamin D raises it considerably, but vitamin D2, if you combine the measurements of D2 and D3, raises it, but nothing like as much as this. And it was only a couple of years ago where the Scientific Advisory Committee on Nutrition actually recommended people take vitamin D supplements, and Sue Lenham-New is on that committee. And it was changed to actually recommend a daily intake of 10 micrograms per day, 40 international units. Before that, there wasn't any recommendation. So this is where I came in. So I said, well, why don't we look at gene expression and see if there's any difference between D2 and D3? But we only had the money to look at 50 subjects at that point. And we were amazed and shocked to discover that those given vitamin D3, the genes which change after 12 weeks, from the first visit to visit 3 is 12 weeks, just taking a very small dose of vitamin D, 15 micrograms, almost all of the genes were different between those that were given D3 and D2, which is unprecedented because it was assumed that they were the same. And the pathways that are affected are completely different. And really key immune pathways are affected by D3. They're not affected when people are given D2. So this is potentially really important. And so we really, this is currently unpublished, but we're fortunate that we can take this forward. So we found that those given D3, obviously the gene expression is very different from those given D2, but the study was actually measuring the different derivatives of D2 and D3 once they'd been metabolized in the liver, and you can see that the actual level of this is 25-OHD3, the actual level of that actually goes down in those given D2, even more so than those which were given no D2 or D3. So we don't know whether these differences are due to the D3 itself or the D2 itself, or whether it's the D2 is actually depleting your natural reserve of 25-OHD3. So it's really important that we determine what that is. Unfortunately, Sue has all these, we have about another 150 subjects, all in the -80, carefully stored for the RNA analysis, because we couldn't have anticipated this result. So we can now go back, because we have all of these in the freezer, those given placebo, and we can look at how their gene expression compares to those given D3 and D2. And that will allow us to tease apart whether it's D2 itself that's causing this difference, or whether it's the depletion of the natural D3. So it's possible that taking D2 supplements actually makes you vitamin D deficient. And so it's really important to look at the label, see whether you're taking D2 or D3. And if you're a practicing Muslim, or if you're a vegan, then you don't get any vitamin D3 in your diet, because that comes, the vitamin D3 in supplements is from sheep's wool, and people tend to avoid that. So they take the D2 thinking that it's equivalent. And this study suggests that it's not. And we're very fortunate that the University of Brighton has provided funding through an innovation seed fund, and Michael and Maureen Chowan have very generously provided funding for us, to enable us to complete this study, to increase the power of the study as well, to include more D3 and D2 treated subjects as well. Right, personal genomics. So, as Deborah mentioned in her introduction, I feel really genomics is going to transform our lives, and I feel really privileged to be living in the era of genomics. And my introduction to genomics and genetic testing was quite acute, because my father developed Huntington's disease. And Huntington's disease is, so this is at my wedding with my two brothers and my father. He was already showing signs. We didn't know he didn't have a diagnosis of Huntington's disease then, but he was showing, it was clear with retrospect that he was showing the signs of HD. And this is a fatal neurological condition with no cure. And It was actually the first mutation mapping to a disease-causing mutation mapped to a specific chromosome, chromosome 4 in 1984, the Huntington gene, and an unambiguous genetic test was developed in 1993. And this mutation, I mentioned that genes are translated to form amino acids. They're translated by reading a triplet code, 3 bases at a time. And there's a segment in the HD gene which has a repeat, CAG repeat. And typically there may be 10, 15, 20, 30 repeats of that in a normal, healthy subject. But if that expands beyond 40, then you're going to develop Huntington's disease in later life, typically in your 50s and 60s. And if it's really expanded, then you tend to develop it much earlier. So my dad had the HD test, and here is his, he's got one from his father, one from his mother. So this is his, from his father, because I didn't say his father died from Huntington's disease. And I should have, I didn't make the point here. HD is particularly nasty because it's what's called a dominant mutant, a dominant mutation. So if the capital H is the mutant gene and the lowercase h is the non-mutant gene, you only need to get one of the mutant genes for it to cause the disease. That's called a dominant mutation. So that means if you're a carrier of the Huntington, the mutant Huntington gene, then every child you have has a 50% chance of inheriting Huntington's disease. But the problem is most people have already had their kids before they know what's happening to their parents. And it affects men and women. It's not a sex-linked mutation. And so this is my mother's, this is from his father, this is from his mother, so this is the healthy one. So I was faced with a decision, and I can only liken it to walking into court to hear if you're going to receive a death sentence and go on to death row with no chance of clemency, because it's the first unambiguous genetic test, so you have to go through a lot of evaluations for that. And I was delighted to discover, so the geneticist from St. George's, they actually got my father's DNA and analysed it and provided me with all the data because I'm a molecular geneticist. And there's my, there's the one I got, the allele I got from my dad, 16, and so that one is the one from my mother with 19. And remarkably, both of my brothers inherited the 16 repeat from my dad, which means all 3 of us were negative, which is a 1 in 8 probability that we'll all be negative. So after that, for me, genetic testing, that's kind of extreme. What really put genetic testing on the map was Angelina Jolie. And the timing of this was a coincidence. This article came out in Time magazine in 27th of May, 2013, precisely when I was at this conference to receive my own genome sequence. I had my genome sequenced in May 2013. And I was at this symposium where every delegate had their genome sequenced. It was a really kind of self-indulgent kind of symposium. And we all got, our rooms had gift-wrapped iPads with a ribbon and a badge saying, "I've been genomed." And inside was an app, My Genome app, preloaded genome, so you could explore your own genome. It's just a fantastic Christmas present. Angelina Jolie, sorry, let's go back. Her mother died of ovarian cancer when she was 56 and was found to carry a really ruthless mutation in a gene called BRCA1. And it's such a ruthless mutation that you have between a 60 to 90% chance of developing breast cancer and a 40 to 60% chance of developing ovarian cancer. So Angelina Jolie, because she had a number of biological children and adopted children, and she'd seen her mother die young, she decided to have the test and on the basis of that have a double mastectomy, and she went public about it. And she's done more to put genetic testing in the public sphere than anybody has. And since then, she's gone on to have her ovaries and fallopian tubes removed as well, because she was showing signs of developing, potentially developing cancer of the ovaries. So at this time, around this time, sequencing has just taken off incredibly. This is when I was sequencing DNA myself, 1980. This is a logarithmic scale. It's just completely gone through the roof, where you can sequence a whole genome overnight now, a whole human genome. In fact, I've got the flow cell that was used to sequence my genome here, if anyone wants to see it later on. That was used to sequence my genome 50 times over, 300 billion base pairs on this little flow cell here. It's incredible. But the technology's moved on so much now that we can sequence single molecules. This is a sequencing machine. You connect it with a USB-C socket. You inject your DNA into the flow cell. It's like Star Trek, isn't it? It's incredible. And a couple of months ago, the chief technology officer of this, it's a spin-out from Oxford University, Oxford Nanopore, published his own whole genome done with this. So these can be, it's democratizing sequencing. People will be able to take these into their, take them home and do sequencing at home in the future. So sequencing has really taken off now to the extent that biology has entered the era of big data. Already we've almost got to the stage worldwide of having an exabyte of genomic data. That's an eye-watering number, 10 to the 18 bytes. And soon it's going to outstrip the requirements of YouTube and Twitter worldwide, just hosting all of this genomic data. So I've really embraced genomics, and these are the genomics facilities that I've established. So I set one up. I was fortunate, I didn't talk about the funding, but I was fortunate to get major funding in Manchester to establish genomics there, and then get major funding in Surrey to establish genomics. And then moved to Brighton 2016 with a generous support from the university. And this is the third and last time that I'm going to get involved in establishing a genomics facility. This was Brighton Genomics in March 2016, a really nicely refurbished, shiny room handed over to me by Andrew Lloyd. And with generous support from the university, we've now converted that space into a fully functional genomics centre. So this is Brighton Genomics, taken from pretty much the same angle. And the great thing about this is we have 2 members of staff who each highly experienced, got more than 15 years experience of genomics in doing all of the wet experiments, Giselda Booker, and Andy Hesketh, who's got considerable experience of doing data analysis. So between them, they can basically help design experiments for collaborators, for people at the university and elsewhere, and then execute them and analyze the data. Because without that expertise, there's no point in having the equipment. So it's wonderful that the University of Brighton has invested in that, because then we have a fully functional facility. And I've already mentioned this, but literally the first genome, it cost billions, and it took over 10 years, but now pretty much about 2 days and about £700 to do a complete genome now. So I've realized I've got 7 minutes left, so I'm going to speed through. So I had my genome sequenced in 2013, personal genome 894, which is the 6 billion base pairs. And we're all almost identical at the DNA level, but each of us have about 3.5 million single bases which are different from a reference genome. So I've got 3.5 million that are different from you. We've all got about 3.5 million. I've got about 60,000 that are unique to me, the only person who has those variants, but that applies to everyone. That's only about 1 in 1,000 bases of our genome is different between us. We consider we have 6 billion, 3 billion, and there's about 3 million different. 1 in 1,000 is different. And these are called single nucleotide polymorphisms. And this is this app from this company where I could look at my genome, I could zoom in and look at specific, right down to the individual bases and see how I'm different from the reference genome. And then I came across Personal Genome Project UK. Well, Personal Genome Project is a global movement. It was started at Harvard, and then it was rolled out in the Stefan Beck from UCL set up PGP UK, and they just got permission to accept donations for their project. And PGP believes that everything should be open. All your data, you don't have to give your name to it, but all of your genome data and your health data should be openly available to everybody. And if everybody did that, we'd have this phenomenal resource for relating genetic difference to health outcome. And so I donated my genome. It went public, and it's completely public, so I'm genetically naked now. This is when I was younger then. And a friend of mine just adapted that. But I got permission from events. Copyright. So I'm kind of the first person who've donated under open consent my genome. So I literally turned up at UCL with a terabyte drive and said, there you go. And they analyzed it. And so they have this fully automated pipeline. They take my whole genome sequence, they put it through every database and generate an automated list of all of the links. And every single one of these variants, they have a RS number, are linked to databases, and you can just click and look at what your risk factors, the beneficial traits, there's a couple of pages of that. Harmful traits, there's like 7 pages of that. And anyone can download this lot if they can't sleep from my website and have a look. And a couple of weeks ago, there was a launch event where PGP developed an app. It was launched at UCL, and I became one of the ambassadors of this genome app. It's now available from iPad. It's available only on the iPad. And this was generously supported by Michael and Maureen Chowan, the development of this. This is an educational app, and 4 of us have self-identified. And so you can click on me and I talk to you, and you can, or you can talk to Stefan. And it takes you through, it's an educational app for like school kids and adults for the interested lay public. It's now available. And it takes you on a kind of accessible journey of what kind of things you can get from genomes and what epigenomes are. Music was actually commissioned specifically for the app, and the musicians have composed music based on our respective genome sequences as well. So it's a multimedia. So this was at the launch, so Vincent actually developed the app, that's me. Stefan is the director of PGP UK, and Afonso, who's got a brain the size of a planet, wrote all the code to take a raw genome and generate an automated PDF with no human intervention. And these reports are all uploaded to PGP website, and anybody can download them. And there were 6 lucky winners. I drew a golden raffle, so this is the raffle ticket. This winner got her genome sequence for free. There were 6 winners at the event. This is all open, so this paper, a very detailed paper on this, it's a research citizen science hybrid project, because just like with astronomy and everything, you're going to need the public to get involved in making sense of this incredible amount of genomic data. You know, there are going to be smart kids sitting around in their rooms, you know, looking in the cloud, looking for correlations between health outcomes and variants. I'm hoping that I'll get emailed by someone from Bangladesh or wherever in a few years' time pointing out something that's... I haven't got time to talk about this, I'm almost out of time, but remarkably, you can actually reconstruct human faces from genome sequence. This is an artist, a New York-based artist, who's 3D printed faces based on genome sequences. This is based on Chelsea Manning, the Marine who underwent gender reassignment, and this artist has been playing with parameters of the DNA sequence to print the face. That's currently touring Europe. I don't really have time to talk about this, but suffice it to say that your DNA sequence is fixed, but it can be modified by your exposure to your environment and your diet and whether you've had traumatic life experiences. And that kind of decorates your DNA with methyl groups, and that affects the activity. And you can now scan the whole genome for the methylome, the epigenome, to see what's happened. And you can predict biological age, and it predicts I'm a male, which is good, but also predicts that I don't smoke. I used to smoke like a chimney when I was younger, but I quit about 25 years ago, and my epigenetic profile for smoking has reversed back to non-smoker, so it's a reversible change. But you can actually, your biological age can differ from your predicted age. So Stefan Beck, the director, he's biologically 6 years younger than he is chronologically. I thought I was going to be 10 years older, but I'm actually the same, which is great. I haven't aged prematurely on the basis of my methylation. I don't have time to discuss this, but it's just Eric Topol, who's an inspirational physician who's really embraced genomics and digital technologies. He has a couple of institutes in San Diego. He was at the meeting, he had his genome done as well. And he talks about the creative destruction of medicine by genomics and all the other digital technologies. And he's published this really accessible book a few years ago on the creative destruction of medicine. And it's now 7:30. So just a couple of take-home points. I think collaboration is so important. It gives you a completely different perspective on science, because so many people just work on their own little thing, and nobody cares, really. But if you start working in a big multidisciplinary team, you can achieve a lot more, like with the work with Sue Lanham-New, bringing together nutrition and genomics, and the sleep researchers bringing genomics into the sleep. And there's a lot of potential in Brighton, the university and medical school in Sussex, and the hospitals to bring genomics into their work. It is a lot of work working across disciplines because everyone has their own language, their own vocabulary. It does take time, but I think it does pay off. And obviously I'm biased, I would recommend getting your genome sequenced. So I've got two quick acknowledgement slides. These are people I want to acknowledge because I would not be standing here if it wasn't for these people. And it's Giselda, who's not only runs genomics, she's my wife. She's put up with me for 30 years almost now. My former partner when I was younger. And David Hopwood has supported me right from the first day I arrived at John Innes, and I wouldn't be here without his support. And Nigel Brown's generosity and altruism as well. It's been wonderful. And David Hodgson, who's here, he's probably examined more PhD theses as external examiner than anyone else in the country, I think. And he's examined most of my students. Thank you very much, Dave. And Rob Ketley was a kind of spotty teenager. We both started at Orpington College of Further Education when we were 16. And he's now, he's retired. He lives in Santa Cruz in California and fishes and is very happy. But he's really, I wouldn't be here without him either, because we chat by WhatsApp video every weekend, and he's helped me also with this presentation and some of the slides. So thanks, Rob, when you watch this. And there's just so many people, hundreds of people that I've worked with, because they've been big collaborative efforts. So I just ran out of time to list them all. So I just said all of my students and postdocs and all of my collaborators and international collaborators. And there's been generous support from the University of Brighton and Michael and Maureen Chowan, and GlaxoSmithKline developing a very good relationship with them. And I think I saw a member of GSK in the audience earlier. And thanks very much to many people within the university who've been really supportive in helping us get started, both from the academic side and the support side. And really thanks to my current collaborators, Byung-Kwan Cho, Stefan Beck, Sue Lanham-New, Malcolm von Chance, and Dave Hodgson, and the team at GSK. And, I wouldn't be here, and Brighton Genomics wouldn't function if it wasn't for Giselda Booker and Andy Hesketh, who are doing a fantastic job. Who've hit the ground running and have already delivered on lots of projects. So may I now introduce Professor Tara Dean, Pro Vice Chancellor for Research and Enterprise, to conclude proceedings.